Hi, everyone. Welcome back to Fragile Nucleosome. I hope everyone had a great summer and uh, managed to get some good science done or some good vacations done. Uh, you may have noticed on social media or in the weekly email that we have booked and released the entire schedule for the rest of this year. So if you're interested, check that out and you can see all of the amazing speakers we have coming up. But today we're just focusing on two people, Grace Bauer and Jen Phillips Cremens. And we're really excited to have them kick off, I guess, this semester of Fragile Nucleosome. As a reminder to everyone in attendance, at the end of the talks, you can raise your hand to ask questions, and we can call on you to unmute yourself if you'd like. Or you can simply type the message in the chat or the Q&A, and we can read it for you. So without further ado, I'll introduce our first speaker, Grace Bauer. Grace started her, I guess, advanced education at UCLA, where she graduated with a bachelor's in molecular, cellular, and developmental bio with highest departmental honors and cum laude. And then, she graduated in June of 2020. So right as COVID was just wreaking havoc on all of society, she drove down the 405 to UCI, California Irvine, to start her PhD career, and she joined Evgeny Khan's lab, where she's been doing some really amazing work looking at long-range enhancer activity. I'm not going to spoil any of the things she's going to tell us now, but I would point out that even for someone in their early career as a scientist, she's already got a lot of mentoring experience. She's won a lot of awards, including a Kirstein NERSA F31, which is supporting her PhD career. And I think you'll all agree when we get to the end of the talk that we're excited to see where she goes from this. So Grace, please take it away.
Thank you. So make sure my slides are going. Yep. So thank you so much for the kind introduction and for the opportunity to give this talk. I love these seminar series and it's really exciting to be a part of it. So again, my name is Grace. I'm in Evgeny Khan's lab at UCI, and I'll be speaking on some of our recently published work looking at long-range enhancer activity. So I think as many people in the audience here can appreciate, DNA is carefully organized within the nucleus in order to facilitate gene expression, broken down into chromosomal territories, which are further subdivided into topologically associated domains, or TADs, through loop extrusion. Within these TADs, we find these individual enhancer promoter loops, and this is what our lab focuses on. And one key fact about these enhancer promoter interactions is that the distance between these elements can vary greatly. Many enhancers work on the scale of tens of thousands of kilobases, which is short range in context for me, because we found enhancers that can operate on the scale of upwards of 1 million base pairs, like this enhancer of MYC, which is nearly 2 million base pairs away from its target gene. And this has led to the question, what might be specific about the regulation of these ultra long range enhancers? Part of the reason this has been a difficult thing to look at is that the classical functional way of testing enhancer activity is through a transgene reporter, such as you can see here, where you place your putative enhancer sequence directly upstream of a minimal promoter and a reporter gene such as LAC-Z. And you can then inject this into an embryo. And this is a really powerful system because you get information about if the sequence is actually able to drive expression, and if it does, in what spatial temporal pattern. But because of the limits of constructs, you have to place the putative sequence directly upstream of the promoter, so you only really get short range functional activity. In order to assay long range functional activity, we developed a system we call enhancer swap, which uses CRISPR and a native enhancer and its target gene as a reporter. Specifically, we chose the ZRS, which targets sonic hedgehog in the developing limbed. And this enhancer promoter pair was chosen for two primary reasons. One, it's long range. It's 850 kilobases away from the target gene in mice. It's even longer, closer to 1 megabase in humans. And secondly, if anyone works with enhancers will appreciate this, it's sensitive. Most of the time you delete an enhancer and nothing happens, but the ZRS is required for sonic hedgehog activation in the developing limb bud, which is required for limb outgrowth, and you can see a normal mouse skeleton stained here. If you have any deletion or disruption of the ZRS, you completely lose sonic hedgehog expression, which results in this very characteristic limblessness phenotype. So this allows us to see, are enhancers able to work over long range by replacing them with the ZRS and simply looking to see if the mice have limbs or not. So to choose the enhancers we wanted to test, we wanted enhancers that, based on all the existing data, should be interchangeable with the ZRS. So you can see here the short range assay data showing that the ZRS drives expression in the distal portion of the E11.5 limb bud. So using other short range assay results, we selected 4 enhancers that also drove strong limb bud expression at E11.5. We further validated that these enhancers were basically accessible and H3K27 acetylated in the same population of cells as the ZRS, along with being generally conserved. The major difference between these enhancers is their distance from their target gene. The ZRS obviously is 850 kilobases away, whereas the 4 enhancers we tested ranged from just over 400 down to 70 kilobases away from their target genes. So we were able to ask, are these enhancers able to work remotely? To do that, we generated 4 different transgenic lines where we used CRISPR to replace the ZRS with each of these enhancers. We then looked at whole mount in situ hybridization staining to look for sonic hedgehog expression in the developing limb bud. And the absence of any blue dots here indicates that none of these embryos express sonic hedgehog. Resultantly, all of these mice had a distinct limbless phenotype, indicating that none of these enhancers are able to activate sonic hedgehog gene expression, and that despite being very strong short-range enhancers, these enhancers are non-interchangeable at long range, and they cannot function when placed remotely from the target gene. So we did several experiments to try to figure out why these enhancers weren't working, and were able to rule out issues with enhancer promoter incompatibility and enhancer silencing. I'm not going to go into the details for this talk, but I can talk more about that if you're curious. And came to the idea of distance, and possibly the concept that maybe some enhancers are just short-range enhancers that don't work at long range. To further look specifically at this idea, we came back to HS72, which was at 400, just over 400 kilobases away from its target. What you would think to be a long-range enhancer. It obviously works over 100 kilobases at its endogenous locus. So looking more at the enhancer at its endogenous locus, you can see here, the orange block is what was originally defined as the HS72 sequence based on what works in the short-range assay. And underneath, I just have the conservation track from UCSC overlaid. You can see the higher the blue, the more conserved that sequence is. And usually things that are conserved are functional. And what we noticed was that there was this heavily conserved region, which was cut off, not included in what works in the short range and what was defined as the enhancer. And we hypothesized maybe this is some sort of range extender element. Maybe this is what is allowing HS72 to work over hundreds of kilobases at its endogenous locus. After validating that this sequence is not capable of activating activity at all inherently, even when placed directly upstream of the promoter, we wanted to see if we extend HS72 to include this putative REX element, does this rescue the ability of the enhancer to function at long range in the ZRF locus? So to do that, we generated another transgenic mouse. Instead of just swapping in HS72, which you can see the data again above, we extended the sequence to include this range extender REX element. And when we do that, we have a rescue of the ability to activate sonic hedgehog gene expression, as you can see this purple dot in the posterior limb, and that this rescue of gene expression is sufficient to drive full limb outgrowth. So this sequence is necessary for long-range enhancer activity. To see if it's also not only necessary, but sufficient, we looked at the shortest range of the enhancers we tested, MM1492. We wanted to see if we could create sort of a chimeric enhancer where we append the REX element. Does that confer the ability to MM1492 to also work over long range? And again, you can see that we, by adding the REX element to the MM1492 sequence and placing that into the ZRS locus, we have a rescue of sonic hedgehog expression shown by that purple dot, which is again sufficient to drive the outgrowth of the limb, indicating that this element is not only necessary, it's sufficient for long-range enhancer activity and can act modularly. You can add this to an enhancer to turn it into a long-range enhancer. To try to understand how this sequence is able to do this, we just actually started by going back to conservation, since that's how we found it in the first place. And here you can see the conserved block is relatively short, it's just under 200 base pairs. And we look to see what transcription factor binding motifs were really heavily conserved within this sequence, and identified two LHX motifs along with a LEF1 motif. To get more of a global perspective, we also took a list of long-range enhancers and looked to see what transcription factor motifs were generally enriched in these long-range limb enhancers compared to short-range ones. And you can see the top enriched hits here. And it started to see a trend where this LHX motif was showing up in both cases. To sort of functionally test which of these motifs was required for the Rex elements activity, we generated two more transgenic lines, which included the HS72 sequence with this extended Rex element, where either the LEF1 motif or the two LHX motifs had been disrupted. And while disrupting the LF1 motif has no impact on the ability of this enhancer to activate at long range, disrupting the two LHX motifs causes a reversion of the rescue, a loss of sonic hedgehog gene expression, resulting in this limosis phenotype, indicating that it is these LHX motifs which are allowing the Rex element to support long-range activity. To further investigate the role of these LHX-like motifs in long-range activity, I went to the most well-characterized of the long-range limb enhancers, which is actually the enhancer we've been replacing this entire time, the ZRS. And then there's been very great work from a lot of different labs characterizing transcription factor binding motifs within this sequence, but we were able to identify three previously unannotated LHX-like motifs, which you can see highlighted here. We wanted to ask, are these motifs doing the same thing they're doing in the Rex element, where they're required for specifically long range, but not short range enhancer activity? To do that, we first assayed short range activity by generating a mutagenized version of the ZRS and just placing that directly upstream of GFP. When we look in the developing embryo, we can see that despite these motifs being disrupted, the enhancer is still robustly capable of driving target gene activity in the correct spatial temporal location. However, when we swap this into the long-range assay, we find that these mice are limbless, indicating that these motifs are required for activation of the target gene over long distance. In summary, we were able to support the model that enhancers are in fact distance dependent. Some enhancers work at short range, but don't work at long range. Furthermore, we were able to identify a novel sequence element that we call a range extender or Rex element, which is necessary and sufficient to confer long-range activity. And furthermore, we did find that the ability of the element to do this is dependent on these LHX motifs inside of it, and that long-range enhancers may have these same LHX motifs embedded into the enhancer, where they're still only required for long-range, but not short-range activity. Finally, I'd like to again thank the organizers for the opportunity to give this talk, along with my lab, all of our collaborators, and my funding sources. Thank you very much.
All right, thank you so much, Grace. That's so awesome. So I guess we've already got a hand raised. So let's see. Oh, the hand raised went away. Okay, I'll go ahead and ask my question first then. So, I mean, it's such elegant work being able to kind of cut and paste these things and make a billion mice to do this. I guess now bioinformatically, can you look at like the population of enhancers that are annotated and then try to identify based on the motifs or other characteristics whether or not specific enhancers have long range capability, and then those may or may not be previously known as being long range enhancers that would then potentially have some long range context in the yet unknown developmental time point.
Yeah, I think that's trying to get right now. I kind of have an N of two, which is the motifs within the ZRS and the Rex element itself. So not really enough of a foundation to do bioinformatically. There is sort of like a grace powered spreadsheet of me manually checking enhancers and being like, based on all the trends I've seen and like staring at this for 5 years, this is what I think might be putative like Rex elements. But they still need to be functionally tested. And I think that's a really interesting thing moving forward, or especially if people can find something similar in other systems, like cell lines where it's easier to sort of do these really high throughput testing. Um, but there are some trends. I think like this, I'm looking now to see like the transcription factors we think might be binding, and then you can use binding data to try to filter out these elements. But that is, and then also elements that it's kind of interesting. We're able to now use when people have done these reporters and been like, oh, this is a negative element, it doesn't drive activity. Now I'm really interested in looking at those because those might be facilitators if they have, this motif in them. And if, maybe if you knock them out in vivo, they actually do something.
Cool. So then I think, Ben, do you want to ask your question first?
Yeah, fantastic talk. I really enjoyed that. Just wonderful work. So my question is, have you looked much at what is happening with the promoter in terms of specificity, in terms of when you have this Rex element and the enhancer, how does it know which promoter to enhance?
Yeah, it's an interesting question. Enhancer promoter specificity is definitely a beast. The thing about the ZRS, there is a gene in the middle, but it is heavily silenced. And there's also CTCF. There's a CTCF loop that supports the ZRS interacting with Sonic Hedgehog. That's actually outside of the region we interfere. So that actually, I don't touch that at all. So these, in the case that we're looking here, the enhancer and promoter are always held very close in contact. It's a completely invariant loop. So in that case, I don't really see any aberrant gene expression with this element, but this may be a method to support enhancer promoter specificity. It's possible.
Thanks.
So then Sergey has his hand raised. I'm going to allow you to talk, Sergey, so if you can unmute.
Beautiful work. So I have a question about what it takes for an enhancer to be long-range enhancer. So you showed that there is a special element with a specific transcription factor. What if it's a thought experiment, perhaps? What if it's a question of a bulk availability of elements along DNA? So if you extend the existing abundant transcription factor, will it become a range extender element? Will that be sufficient? Or there's some special range extender transcription factors whose main function would be range extender. So is it simple or is it complicated?
I think it's likely both. And I think some of it is an interesting case of in the NF2 I've looked, both of these clusters of motifs are on what would be considered like the edge of the enhancer. So there may be an element there where if you look, it's depending on where you draw your cutoffs. But as far as I do think there also is something unique about this class, because what's really interesting about these LHX factors is that what they're known for is being cofactors of LDB1, which is, if you've read like some of Stavros Lombardi or Bobo's recent work, is a looping factor, one of the only really well studied looping factors other than CTCF and cohesion that can actually sort of anchor two disparate regions together. So I think that's really interesting. So it may be a combination of sort of both models.
Awesome. So then we have a couple of questions in the Q&A. I'm going to go ahead and ask the first one. Sachin Pundir asks, do you think there's any similarity between the Rex motif and the Waisaka lab coordinator motif?
Yeah, I did. When they had their preprint, I was trying to see if it matches. It does match a lot. There's no, I was trying to remember, I couldn't find any twists, which is what they think is binding to that in the limb. So I don't know what specifically is binding there. So it's a possibility, but I do think it might be other factors. So I think it's probably more likely to be LHX2 and LHX9.
I guess that's what I was just thinking of is the motifs in the range extender, are those dependent on what transcription factors are expressed, right? Like if you have
yeah,
a long
so
range,
when I was doing the motif doing, when I was looking at motifs, the first thing we do is do filtering. So these are all limb. I cut down to limb express factors because like that's the thing with the LHX motif is it's a family of like 12 transcription factors. That's part of the homeo box, which is like several hundred. But we're filtering down to the ones that are actually expressed in the ZPA.
Right. So do you think it is like maybe more generally like organism wide, it can be homeobox factors that have this function, or do you think it's something special about LHXs?
I think it might be something special. I think it might be LHXs partially because we do see a slight preference towards specifically the LHX family of at least on the motif level. And then the other thing is, even though this is a smaller subset, this is actually an extra slide that I had where, at least in development, these are a really intricate pattern across a lot of different tissues. They're master transcriptional regulators in a lot of tissues. So I think there might be a model here where this is functioning across development. And this might be a way of activating specific long-range enhancers.
Awesome. Well, Grace, thanks so much. You have a few more questions in the Q&A. If you don't mind, you can answer those while Jen is talking. But beyond that, I will pass it over to Ben to introduce our next speaker.